Hello, everyone, and thank you very much for joining. Welcome to today's webinar. The title of today's webinar is Structure-Based Drug Discovery Without a Structure, Enabling FEP Prediction for Challenging Targets and ADMET Anti-Targets. So my name is Jeremy Van Dom. I am Director of Application Sciences and the Head of Research Services and Research Enablement Services at Schrodinger. Our other speaker today will be Edward Miller, the Senior Director of Protein Structure Modeling at Schrodinger. And I encourage you to use our Q&A feature to ask questions throughout these presentations, and we will answer as many questions as we can at the end of these presentations. So to set up the stage for today's webinar, we need to start with the free energy perturbation, or FEP technology. It's at the same time the most impactful structure-based approach that we have, and the most challenging in terms of what it requires in terms of structural data. And it is so impactful that it can be game-changing, and this is what motivates us to push the envelope in terms of enabling more and more structures to be used for accurate FEP predictions. So just in a few minutes, I want to give you a sense of the level of accuracy that you can expect from FEP and what we call the impact of FEP. But I'll start by saying that since our first release of the FEP implementation, FEP Plus in 2015, 8 years ago, the method has been widely adopted across the industry, and you can find out there a lot of publications by partners or collaborators, or just people who have independently tested the tool and verified its track record in terms of accuracy and impact. So we're happy to share the list of these publications, but I'm sure that you might have come across some of them. What do we call impact or accuracy of FEP? So first, just a quick illustration of what FEP is. It's a rigorous physics-based method that starts from a structure of the target protein with a ligand of interest, the specific series that you want to study. It is an all-atom simulation. It includes explicit solvent waters. And what we do is really, we take a first molecule, presumably a reference molecule for which you have experimental data, and we alchemically modify this molecule by introducing a chemical transformation. In this case, the hydrogen is changed into a chlorine, and we rigorously calculate every single term of the binding free energy, and you can see that we can have a prediction of how much this chemical change will impact binding free energy. And today, this method has been tried on hundreds of targets. So I'm pointing you to a pretty old paper because this is what you have access to. But today, it's been tried on hundreds of targets. And the average error that we observe when the system is properly enabled is clearly less than a pKi unit, around 0.7 pKi unit, about 1 kilocalorie per mole. And to go one step further in terms of understanding what this accuracy is, we went through the exercise of identifying out there data sets where the same compounds had been measured experimentally for their either KD, Ki, or IC50 by different teams or independently in different experiments. And we could identify 60 of the data sets. There were about 50 targets and more than 1,000 ligands. And that allowed us to create this little profile of the experimental methodologies error throughout these different data sets. And as you can see, FEP's error profile on the same data set is slightly shifted upward, as you would expect, but not so much. The profiles are comparable. So in many ways, at least internally, we do consider FEP as an in silico assay, right? And to finish on FEP and go one step further, I wanted to illustrate on these very simple I would say, illustration what it means to have a project enabled by FEP. On the left-hand side here, you have the pKi, or the potency of a number of compounds that have been synthesized across the life of a program, about 2 years and about 1,400 compounds. And anything above 18 pKi is considered to be meeting the project criteria. And what you can see is that we started with a good compound, about 10 nanomolar, and as we tried to optimize other properties, we lost potency, and we need to work our way back up to the on-target potency that we desired. And that happened again and again, to the extent that if we consider all the molecules synthesized, only about 10% were meeting the on-target potency criteria. Now, fast forward 6 years, when we did have FEP and we started the same project again that we had to stop because it was hitting a wall, we started with some virtual hit screening. You can see potencies around the micromolar range, as well as a new core that had been identified through FEP core hopping, which is a different topic. But what you can see importantly is that we could maintain the on-target potency throughout the optimization effort of other properties. So about 66% of the molecules synthesized were meeting these on-target potency criteria. That is pretty much what FEP allows you to do. It gives you an unprecedented control on-target affinity. So going back to the topic of this talk today, and realizing that FEP can potentially be a game-changing technology for a given program, we really wanted to enable as many FEP projects as we could. The challenge is that typically, in order to have FEP enabled, you need very good structural data, and specifically atomic resolution X-ray of the target in complex with a specific series of interest. And of course, you have a lot of other cases. It goes from no structural information on target at all, or insufficient structural data, low resolution cryo-EM, or the structure of the target with a different series, or APO. In order to help and be able to enable FEP in these cases, we developed a number of technologies. Some had to deal with structural refinement, and it's not the topic of today's talk, but GlideEM and Phoenix OPLS are two of them. And some others are aiming at generating and identifying the best pose, including with protein motion, induced fit motions, and IFDMD is one of these core technologies that have been developed exactly with this mindset. So we use these tools and acquired quite a bit of experience doing just that, enabling FEP in the absence of optimal structure when we're not in that case. And we've done that in two contexts, internally in the Schrödinger's therapeutic group, and we have a number of examples there, but more recently also through services, which we call research enablement services. And we'll illustrate what type of systems we've been able to work on. And more importantly, what you can see already on that slide is that the starting points can be very different. We have had success all along the scale or the spectrum of structural information available. And I want to start with a validation study where we could enable FEP for a GPCR starting from an AlphaFold structure, because this study is, illustrative of the approach, and it brought up a few lessons learned that are important. I want to start by saying that we know that AlphaFold structure are not right away enabling FEP. First reason is that a few of these, certain regions are low confidence regions, and the most important reason is that there is no ligand in AlphaFold structures at the moment, and we do need this complex structure. So Ed's team, the next speaker's team, has made a case study on the somatostatin receptor. This study is likely to be published soon. You can already have access to the manuscript here, and we'll go through this study right now. So when this study started, there was no experimental structure for the somatostatin receptor family, none of the members. So since then, though, SSTR2 and SSTR4 have been solved, but these data have not been used for this effort here. The closest homologue at the time were opioid receptor with around 30 to 45% sequence identity. In order to enable FEP in the absence of structure, we still need to start from something we know, and this something we know is structure-activity relationship, SAR. In that case, we used a BI patent. It was an agonist series of about 79 ligands. What's notable here is these were cell functional assay data. So there is a little extra noise that we can expect from the data, and yet sometimes this is what we have to work with. I want to introduce here already that we could distinguish two subseries in those 69 that we picked, one with a 6-6 double ring here, or fused rings, and another subseries with a 5-6 here. It doesn't really matter, but what matters is that we could split the data set in two subseries. And you can see the dynamic range is pretty large. That's ideal. The larger the dynamic range, the most information is embedded in the SAR, the better for what we want to do. So the approach is the following. We started with a number, 5 in that case, different AlphaFold models. And you can see the overlay here of the AlphaFold models. They are quite similar. You have a few loop variation. So the loop is not right away in the orthosteric side, but you don't know what effect it might have, and some small helix motions. At first sight, these different structures could appear, or the difference might seem non-significant, but we'll see that they can be significant. The second step is that from each of these 5 models, we used induced feed docking, this IFD method, and obtained 5 binding poses per target. So to say a few words about IFDMD without going too much into details, we can illustrate here in this case where we happen to have an X-ray structure for both the apo and holo structure of the same target in complex with the ligand, that if we were to use regular docking on the apo structure, we would never find the right binding pose because of some side chain clashes with the ligand. In those cases, what you need to do is to have some induced feed, I would say, captured by the docking tool. And this is what IFDMD does in a very successful way. And you can see that the pose in that case could be found and the side chain could be reoriented in a more favorable way. You have more details in these publications, but for our purpose here, what really matters is that from each of these 5 AlphaFold models, 5 very reasonable poses with induced feed effect could be created. So we've explored both on the protein side and on the ligand side the possible conformation, and that gives about 25 target ligand complexes that at this stage should be evaluated. And our way to discriminate between these different target ligand complexes is their ability to be predictive when you start FEP with them, or when you use FEP. So just as an illustration, on these plots, you have the experimental binding free energy. It could be the pKi on the x-axis, and the predicted pKi or binding free energy on the y-axis. And what you see is that some models are clearly not predictive. There doesn't seem to be a signal, and some others will show a signal, will show a good correlation between the predicted binding affinity and then the experimental one. The metrics we use to capture that are the R-square, the correlation, but arguably more importantly, the accuracy, the root mean square error, how much deviation is there compared to these diagonal here. And the underlying hypothesis of the whole approach is that since FEP requires to start from a good structural model in order to be accurate, if you are accurate using a certain model, this model will be close to, will be correct or close to the native one. So in practice, this is what it looks like. We applied exactly this approach, meaning assessing the 25 models, so 5 AlphaFold, on which we had 5 different IFDMD poses, with only the first subseries for efficiency reasons. That's a first scan, and we want to see if we capture something, but we can do that with the subset. And that allowed us to identify or focus on 3 specific protein ligand complex structures or models, where the R-square was, not too bad. The closest to 1, the better, of course. And the root mean square error, more importantly, was close to the optimal one or the expectation, which is about 1 kilocalorie per mole, as I presented initially. And these 2 models are doing really well. And this third model here is mediocre, but it seemed worth to be salvaged. At that stage, we used the second set or the rest of the SAR, the second subseries, to see if we could further discriminate between the models. What happened is that the 2 good models remain good, so we could not discriminate further these 2 models, and the mediocre models stay mediocre. At that stage, it was clear that it could be abandoned. And moving forward, we used ABFEP in order to try to discriminate between these 2 models, and we're successful in doing that. You can see that the first model, coming from the 5th AlphaFold structure and the first IFDMD pose, gives a binding affinity prediction from ABFEP close to the experiment. I should say that ABFEP is a methodology derived from FEP, but that allows to not need to have a reference structure to start from. So this is why we can get to an absolute number here. And now, looking at the final result, this is the type of correlation we could obtain in this specific case. You can see that most of the compounds are in the dark gray area. The RMSE is very good, 1 kcal per mole, and the R-square is very reasonable too. And you can see that a model like that could very well be used prospectively and be useful in a program. And of course, we have all the details of the binding mode that can also help to ideate with intermediate waters, etc. So now, these specific case studies is illustrative of the whole approach that we want to have. And we have a few lessons learned that are important. The first one is that small difference, easily overlooked structural differences between models can have an impact on FEP. So the cyan pose here, or the cyan model, is the best one that I presented just before. You can see the statistics here. They are very good. Now, the pink one was also among the 25 models that were assessed. It was actually the third one, the mediocre one. But you can see that the FEP accuracy is significantly degraded. And going back, the team could identify that this was due to a very small difference on the in one specific helix here that was pushing these tyrosine a little bit further in the pocket and in turn pushing the compound a bit further deeper in the pocket. And that prevented a proper interaction with this serine here. This is something which in hindsight seems clear and we can understand, but it's very hard to anticipate, to predict as a critical structural difference. And that's the second, that's another lesson. It can be worth, it is worth in many times starting from different starting points. And seemingly similar structures might have small differences that are only going to be revealed by the end. And so the second lesson was that some but not all of these structural details can be resolved by IFDMD. In that case, for example, IFDMD could not retrieve exactly the blue pose starting from the pink protein conformation. In the end, the final lesson for us is that it's very powerful to combine multiple starting points, multiple target structures with IFDMD. And I'm going to take you now to the methodology. We have consolidated a global approach for this type of problems, and it's a busy slide, so I'll take you through it. But the high-level lessons are already, described to an extent. So it's basically a two-step approach. The first step is we generate an ensemble of models, and we basically go large scale. And what we call large scale can be, a few models to a dozen to a couple of dozens of models. We've gone as far as doing, 40, 50 models in services specifically. And you do that by combining different starting point with IFDMD. The second step is, out of these models, we need to, as efficiently as we can, converge towards one that is predictive of, or is predictive enough to be used prospectively. And this is done in a stepwise manner using trial and error with FEP. And the type of things we do in order to be efficient are to start with a well-defined subset of SAR, and one example is SAR focusing on a specific region of the ligand, so that the analysis of the first rounds, let's say, is as informative as possible. If a model does well in a specific region, it probably means that the model is not too bad in that region, and we can use that as a starting point to optimize the model. And you can see that after step by step, we can eliminate some models, regroup some others as being very similar, and slowly converge towards a model that can predict the global SAR. So depending on the case, it doesn't have to be the case. You can be very quick in finding a model that is predictive with FEP, but it can involve a significant effort, at least computationally. I want to say that, as I mentioned before, quite a bit of experience has been acquired recently through services, and we offer that because it's, again, potentially very impactful for people, and at the same time could be difficult to deploy for a customer because it demands quite a bit of computing resources and licenses. We offer that as a service, and this is something we're happy to engage with you on. But through all these services, we've had quite a bit of success, and I want to give you a sense of the type of targets we've worked on and the type of things we've discovered. So I have a few last vignettes before I pass I give it to Ed. So the first case I wanted to talk about is the one where the team we worked with started with a cryo-EM structure, but the project was in effect not structurally enabled, and that was due to the fact that they tried to to explain the SAR they had with this cryo-EM structure, and they couldn't. They were expecting something very specific. There is a piece of SAR where they were hoping to explain, and they couldn't from the cryo-EM. So they knew something was off. Yet the resolution was not too bad. They had some SAR, and quite a bit, 70 compounds, a good range. And the approach we had was to start from, of course, the cryo-EM, but also a few different, slightly different structures coming from the refinement of the cryo-EM in different pieces. And we deeply explored also the ambiguities of the density. We went back to the density and identified regions where the model was made for and delivered in terms of a cryo-EM structure, but it was not clear that things were as they were modeled initially. What we found in the end, and you can see here the final results with 33 compounds, an MUA of below 1 and RMSE of 1.2, which is quite good. We found two things. The first one is that in order to be predictive, the binding site needed some rearrangement, and there was some slight but significant. Some helices needed to shift a little bit, and refinement was not enough. The second note or takeaway from this project is that the ligand binding mode was counterintuitive. It was flipped compared to what the cryo-EM model was saying, yet it was fitting into the density, not as well, but it was fitting into the density. And very importantly, it ended up explaining SAR, including some new compounds that came up afterwards, and some puzzling parts for the team. So one lesson for us was beware of intuition, because it can be misleading, and empirical data can be very useful. So the second example I want to take you through very quickly here is that of a kinase. And we tend to think of kinases as cases, easy to solve. You have a lot of templates out there, and a lot of binding modes also known. This case wasn't easy at all. You can see that the accuracy was okay, but a few outliers remained. That's another interesting point we can take across, is that depending on the amount of structural data the team has at a given time, this effort might only be able to take them so far, and you might end up with a couple of candidate binding mode that you need to further discriminate using prospective approach. But in that case, we could end up on something reasonably predictive. And the approach used was, we explored 22 protein ligand complex. To give you a sense, we used homology models as well as AlphaFold, multiple ADMET pose each time. And the final pose revealed important by its effect, but small in amplitude, structural reorganization of loops and side chains in the binding site. And interestingly, in the same project, we were able to enable an anti-target in the same family, a kinase, for the same series. And what we found was that the binding site was not the same in the target and anti-target. It was basically flipped or shifted, and flipped, if you will. So again, it was counterintuitive, but the data was clearly speaking. And maybe I will finish with this one, which was a real case, GPCR enablement. Again, a case where the team felt that they were well positioned to enable their program, because they had two proprietary X-rays with different subseries, granted, but two proprietary X-rays of relatively good resolution. There were two cryo-EM structures in the public domain, and they had quite a bit of SAR. It wasn't an easy effort, but you can see that the final result is very, I would say, satisfying. And with a model like that, the team felt confident that they could move forward with prospective application. I would just say again to give a sense of the type of effort that is, and the broad range of cases. That was a case that was harder than anticipated because again we started from X-ray structures of the target with a different series, but still, the first effort that was quick led to a model that was clearly capturing some signal, as you can see. But the statistics, the accuracy, and the R-square was very suboptimal. And in order to go from this model to this model, and there is a significant difference in terms of usability, possible impact between the two models, we actually needed to go pretty deep into understanding exactly what was driving potency in this model. So 36 extra models needed to be built, and some specific local optimization need to be made. But, and that's the last point I'll make, that was very important for the team, and they decided that it was worth investing the effort. And the effort usually leads you to the type of accuracy you want if you have enough structural data, I mean, but if you decide to go down this path, right? And you can see that with 75 compounds, we now have a much better correlation than we had before. So I'll let Ed take over from this point.
Hi, everybody. Thank you very much, Jeremy. I'm Ed Miller, Senior Director of Protein Structure Modeling. In the vignettes you heard from Jeremy, the story was very much dominated by retrospective work. In other words, here's a data set. Can we provide the customer and ourselves a structure which could explain the known SAR? And that's great. And we have the expectation and a few bits of anecdotal support in those cases that if you go and advance things to new compounds, that these models hold up. But how far can you take it? What is the evidence that these models built from retrospective data can be used prospectively? At what point do they start to fall apart? How much data do you actually even need to produce a good retrospective model? So we can answer some of these questions based on what we've done in our active internal programs. And I'm going to explain this within the context of some very difficult anti-targets, these ADMET anti-targets, 2D6, 3A4, and hERG. So let's jump right into that. So what I'm showing here was a program which discovered rather late in the game a specific inhibition of cytochrome P450 2D6. So this is an enzyme that I'm sure a lot of folks here are familiar with that's involved in metabolism of drugs. And so if you inhibit it, you'll cause a lot of drug-drug interaction. So what was seen here was that the inhibition was specific to a particular isoform of cytochrome P450. It was the 2D6 isoform. So it wasn't 2C9, wasn't 3A4. And that was very interesting because this is apparently a very specific effect going on, not some broad. What was also seen was that among a number of compounds, there was sharp SAR. Now, this is an active program, so I'm not going to be able to show you the full compounds, but going from compound 2 to compound 3, there was this 10-fold drop in inhibition, and it was due to very small changes, which really was not understood. This was late in the game, I mentioned. The team had optimized these compounds for on-target potency to have a lot of other satisfactory properties. They didn't have the luxury of making broad changes without having to go back to the drawing board and destroy a lot of their hard-earned gains. They wanted a model which could allow them to make very small, very precise changes to get rid of this 2D6 inhibition. So let's start with that. How are we going to model that? Well, I'm showing here on the right the 10 public 2D6 structures that contain a ligand and are not mutated. These are all wild type. And you can ask the question, If you were to arbitrarily pick any one of these 2d6 structures, could you successfully get your ligand docked in, even using our induced fit docking technology? Now I'm showing here among the 10 structures, there's a fair amount of receptor motion. You can see there's this loop motion here, there's this helix motion going on up here. And so can you just arbitrarily pick any one receptor and hope that the induced fit docking technology can get you to where you need to go? And the answer is, in a prospective program, you don't really know. I mean, maybe in some cases, yes, but you don't want maybe to be good enough. You want as much certainty as possible. This is a real-life program. Time and money is on the line. So what we did was we did the most robust thing. We docked a project compound into all 10 of these public structures. But there's an extra wrinkle. The ligand has a sulfonamide which could exist in either the neutral form or negatively charged. In the on-target, we knew what state it could be in, we knew which state mattered, but in the CYP, who knows, could be either of these states. The induced fit docking algorithm, IFDMD, it doesn't vary the protonation state of the ligand. What you supply is what you keep. So what we're going to do is we're going to run independent calculations. So we're talking independent calculations where we dock into the 10 available public 2d6 structures, and we're also going to vary in these independent calculations the protonation state of the ligand. What we were talking about ultimately were 60 models that we had to interrogate. So we're going to interrogate it in all the manners that you saw Jeremy showed a few moments ago. We're going to use free energy perturbation. And at the time, the project team had only assayed 10 ligands from this series and got IC50 data for these 10 ligands. So we have quite a lot of models and a fairly small amount of data. How does this perform? Well, what it looked like was that we had 3 models which gave a signal, but remarkably, these 3 models looked very different, and we can qualify how different they look based on their coordination to the heme or lack thereof. So for example, on the left here, we have a model which has no coordination with the heme, and its FEP performance looks quite good. In the middle, we have a model which has a water-mediated heme coordination, and yet remarkably, with the same 10 ligands, the FEP performance is still quite good. An RMSE under a kcal, an R squared of 0.75, and then there's a third model that has direct heme coordination, and the results are still good, a 0.69 R squared, an RMSE of a little more than a kcal. So which of these 3 models is correct? And the answer is we need more data. But having 3 models as opposed to 60 or as opposed to no models, you can rationally decide what changes you want to make to try to break this apparent degeneracy. Add changes here where one model says that helps and another model says that hurts. Then you go and you assay these compounds and you see which models indeed survive. So that's what was done. We had 10 ligands here with 3 models. If we took it up to 14 ligands, only 1 model survived, which is this one here. This is the retrospective performance. So we were seeing here about R squared of 0.5 and RMSE of 1.5 kcals, and that RMSE was being dominated by this one outlier. Okay, so this model looks plausible, it has a few warts with it, but can it hold up prospectively? So we put it down to this one model, and the team said, if we're going to use it prospectively, let's start to create some ideas around what this model is telling us. And what was done was they examined the model, and if you recall, I said that we had the protonation state of the sulfonamide as something which could change. So in this model, in the model which worked the best, the sulfonamide was neutral. And that was very exciting because what was seen was that this neutral sulfonamide was making a charged hydrogen bond in 2D6 to this glutamic acid. And the team said, oh, this is great. We don't really need the sulfonamide to be neutral in the on-target. We can satisfy our on-target potency if the sulfonamide was negative. And if this model that we see before us is true, surely putting a negative charge next to a negatively charged glutamic acid, well, this thing shouldn't bind. And therefore, if it doesn't bind, it shouldn't inhibit 2D6. So the team decided to make changes adjacent to the sulfonamide to modulate the pKa and shift the sulfonamide to be more negative. That was the idea. That's where we led to compounds being synthesized with that idea. And this is how it turned out. So here we have, this was the original compound, which had good on-target potency, but was terribly inhibiting 2D6. Then we made some small changes adjacent to the sulfonamide, adding a few heteroatoms here and there. This was setting us in the right direction. We preserved on-target binding. 2D6 inhibition had gone down. And then we advanced this even further, and finally we have a compound which was just off the assay for 2D6 inhibition as predicted. And what was nice was this whole time we were predicting the pKa of the compounds. This was using Jaguar, our quantum chemistry package. We were also then, after predicting these compounds, assaying the pKa very simply, and we were seeing that the pKa was in agreement. So we were really testing two things here: the quality of the model that you saw before you a moment ago, and of course our ability to predict the pKa. So with this whole rational understanding as to how 2D6 was binding, we de-risked this liability. So that was 2D6. That's an important target. But if you're familiar with cytochrome P450, the big beast is 3A4. 3A4 is very common in metabolizing more drugs, but also if you inhibit it, it's an even bigger problem. And the other concern with 3A4 is it is far more flexible than 2D6. So how does this problem grow with a more challenging target like 2D6, or anti-target, I should say? Well, we wanted to investigate that first retrospectively using public data. So we took this paper from JMedChem, and it was talking about antagonists to this GPCR, and that's fine, but what we cared about was how the paper talked about how they dealt with inhibition of 3A4 and also 2D6. So the paper goes through a lot of explanations. It talks about all the different things they synthesized, and what was very nice was it contained a series of 47 ligands, each with very small changes, then the associated IC50 data against 3A4 and 2D6. They made these changes, as far as we can tell, without the benefit of a structure. Could we create a structure to explain what they did, to rationalize it? So here's what the ligands look like. These are not trivial molecules. They had a lot of rotatable bonds. They had two positive charges. And what was nice, however, was it was still a good amount of data with a nice wide activity range. So you went from the least inhibiting compound to the most inhibiting compound in 3A4. You were talking about a 4 kcal activity range. Now, there's a number of 3A4 structures out there in the PDB, for better or for worse. There's about 70 of them that are wild type with a ligand bound, a drug-like ligand. And we asked the question, could we pick any of these 70 structures and start with that? And the answer apparently was no. So this here is a heat map that's comparing Tadamoto similarity. And the more red this map looks like, the more similarity there are between pairs of PDBs. This column here on the right, this is one of the ligands from that JMedChem paper. And you can see from the very light coloring here, there was no existing public structure, there still is none, that looks anything like the ligand in this paper. So there was no obvious starting point. So we're going to have to dock into all 70 structures. And that's sounding a little unpalatable. You're going to be swimming in a sea of models here. So we engaged in a assumption, a hypothesis, that the right binding mode is one which is seen independently for different members of the series. In other words, that the ligand series is binding the same regardless of which ligand you put. That is the same in 3A4. So if you enforce that assumption, you can throw away the vast majority of models. So to be precise, when we docked in 2 or 3 ligands into all of these 70 structures of 3A4, we had thousands of poses. But if you ask the question, how many of these poses place the core in the same position for each of these 2 or 3 congeneric ligands, only a few models remained. We took these few models, we ranked them with one of the terms in the IFDMD scoring function. We said, let's run FEP on these few and see if any of them light up. So here's the FEP performance. Only a single showed a signal. This is the only one with an RMSE of under a kcal and R squared of 0.66. So with this amount of data, there were no peer models. There were no apparently degenerate models to compete with. Here's what the correlation plot looked like. These horizontal lines at around -6 kcals per mole, this was the top of the assay. This is where the assay really couldn't tell if it was binding anymore or how weakly it was binding. For example, we're predicting that 2 of the ligands don't even bind at all. But of all the ligands which had a quantitative IC50 data, this model was getting those spot on. And you can see there's just one very small outlier at around 1.5 kcal. The overall statistics, though, was that this model is explaining the SAR to within an RMSE of less than a kcal, 0.9 kcal. So we did the same thing with the 2D6 data. We did the independent modeling. We assumed a consensus binding mode, that the ligands are binding the same way for multiple members of the congeneric series. Only a single model survived. And this is the performance of that model. It's a little less precise than the 3A4 model. We're around a 1.34 kcal per mole RMSE, but surely still good enough to distinguish between ligands which inhibit and ligands which don't. So that was 3A4. That was retrospective. How does this work prospectively? Well, in a prospective case, what we had to work with was percent inhibition originally. So I'm sure this is common among those of you in the audience. The first time this liability is discovered, it's done with a single point calculation. So you get percent inhibition. Remember, though, FEP is predicting binding affinity, which is kcals per mole. And you can convert that into an IC50. How does percent inhibition compare with that? So we had to engage in a simple assumption, one which we know isn't quite true, but is it at least valuable? And the assumption we said is, can we correlate percent inhibition with IC50? And that's what you're seeing here. We just created a linear correlation. Even though we know the relationship is probably more sigmoidal, how many models would remain if we assumed this linear correlation? And then we could narrow things down and take it from there. So we did the same approach I showed prior. We docked into all these 70 some odd structures. We narrowed them down to only pick the binding mode, which was seen independently across multiple ligands of the series. And then we challenged them with FEP using this linearly extrapolated IC50. What we had were only 2 models which remained, and both models looked pretty good except for but one point here. This is what we call the model selection data point. So at this point, we were able to go back to the team and say, hey guys, do you mind spending the extra time and money and getting the actual IC50 instead of using this percent inhibition? And they said, sure, that's a wise use of money. So they went and they did that, and model 1 looked even better. So it went from having an error compared to our prediction was an error by about 1.5 kcals. Once they got the actual experimental IC50, the model even agreed better, said the error was about 1 kcal. And so all the compounds looked good. They even said, hey, here are 4 new ligands we hadn't previously shown you. We added them to the model. They held up. And then model 2, the competing model, it didn't look really very good. You still had that one giant outlier. So here we had this model on the left with retrospective performance at about 1 kcal. How does this work now prospectively? Well, this model has been still used actively in this program. And as of mid-July, there's been 13 compounds which have been prospectively synthesized and assayed using the model. And as you can see, it's still really holding up quite well. An RMSE of about 1.2 kcal, R squared of 0.52. So as far as we got a model here, which makes good structural sense to explain 3A4 inhibition. So let's now move, we're getting a little short on time. Let's jump right into hERG, another common problem. So you don't want to inhibit hERG either. We took the same approach. We had this paper from Bioorganic Med Chem Letters. It was folks talking about antagonists to another GPCR and how they were dealing with a hERG selectivity problem. The paper was very nice. It had 31 compounds. 8 of them had an ambiguous protonation state. We didn't want to have to simultaneously, if possible, we wanted to avoid having to both predict the pKa in the receptor and the binding mode. So we excluded them, and we were left with 24 ligands, a decent set, with a 3 kcal activity range. Here's what the ligands look like. There's this nice conserved core with changes on either end of the molecule. Here's the positive amine that, if you're familiar with hERG, is likely to give you your source of a hERG liability. Obviously, the team couldn't get rid of this positive charge without hurting on-target potency, or they would have, and there probably wouldn't have been a paper to talk about here. So the paper was very nice, but it was also very short, and it had this little sentence in the conclusion saying that they made changes to the lipophilicity and serendipitously found a compound which had low nanomolar activity. It was potent against the on-target, but it was not very inhibiting of hERG. And the question is, do we have to really rely on serendipity when these problems arise? So we said, can we create a structure that can explain what was going on here? We did the same approach for HERG. There's very few structures out there to work with. There's about 3 in the PDB. There are these poor resolution cryo-EM structures, but we went through the same approach. We're going to independently dock 3 ligands. We're going to ask, how often do we see the same binding mode for these 3 ligands? Only 9 models satisfied that. We said, let's run FEP with the 9 models. Only 1 model showed a signal. It had a sub kcal per mole RMSE, an R squared of 0.65. Here's what the correlation plot looked like. One outlier here among one of the most potent ligands. We said it was more potent than experiment, but most importantly, the weaker ligands, which is the ones that you want to get accurate since you're not interested in binding HERG, you want to de-risk HERG inhibition. We were getting these right. Here's how it looked. So the model was placing the positive amine surrounded by these tyrosines in Phi. If you're familiar with HERG, this is the expected location for the positive charge to have a lot of pi-cation interactions. We didn't impose this requirement on the model. It happened to be the one which performed the best, so that was a nice, happy coincidence. And you can see here an overall view of HERG and the location of the ligand in relative to the channel. So this is also the typical expected place where HERG inhibition was going to occur. There's no good structure out there of really any inhibitor bound to HERG. So, we present this here sort of as a benefit to the community. At least what we can tell from our evidence, this is where these ligands are indeed binding. So that was retrospective. That was using a paper out there in the literature. We had a program which ran into this problem and we wanted to solve it prospectively. Same approach. We took 3 ligands from the program. We docked them into the 3 receptor conformations using IFDMD, using induced fit docking. And then we were left with 9 models ultimately to challenge with FEP. And at the time, we had only 9 ligands which had IC50 against HERG. Not a very huge data set. This was the best model. It looks pretty good, we think, an RMSE of under a kcal. The big question, of course, is 9 models good enough to make a determination? Well, this model still held out as the best model. We didn't really have any peers, so we said, let's roll with it and see how it goes. The team said, well, here's an additional few ligands. Here's another 14 ligands that we held out, that we didn't tell you about. Let's see how the model does with them. So those are what have been added here. So we got a total of 23 ligands. And the plot here is pKi as opposed to kcal. So the weaker ligands are going to be shown on the left. And what we were most excited to see was that there were 3 ligands which experimentally were considered to be extremely weak off the edge of the assay. FEP said in this model, these are also the weakest ligands. Now, the overall performance was a little worrisome. The RMSE was fine, 0.8 log units, but the R squared was poor because of these outliers. Now, these outliers were in some of the more inhibiting ligands. So the team was concerned, but at least it was getting the weaker ones right, which is the region of inhibition that the team was most interested in, of course. Nonetheless, they said, let's go and see how this works prospectively. So they said, let's look at the model and see if it can tell us why things are weak and why things are strong. And there was a very nice match pair. So it's an active program, and unfortunately, I have to blind the vast majority of the ligand. But what was seen was that you were getting about a 50-fold drop in potency if you broke a hydrogen bond to a backbone carbonyl. So there were some changes adjacent to this NH that could explain why the hydrogen bond couldn't be formed here. So they said, okay, this is a reasonable hypothesis. Can we use this to de-risk HERG inhibition? So 14 compounds were synthesized in assay using this model, using this idea, and we were happy to see that all 14 ligands were within 1 pK unit of experiment. So these predicted compounds, these are the ones in light blue. And what was, of course, most exciting were there were 2 ligands, which were also predicted to be extremely weak. And we confirmed experimentally they were indeed extremely weak. So this model is now being actively used in the program. They've got their on-target model, and then they have their HERG model. And every time they make a new compound, they can run FEP in the on-target, they can run FEP in HERG, and they can see if it's worth synthesizing. So you can see here the common theme. It's not taking a whole lot of data, 15 ligands, 14 ligands for 2D6. For HERG, it takes 9 ligands. It doesn't appear to take a whole lot of data to produce a model that is satisfactory. As far as we can tell, there is no other method out there, especially a machine learning method, which can work with such a small amount of data and give you a model that is so general. Now, this is something that you may be interested in internally at your company. Let me turn this back over to my colleague, Jeremy, to explain how we can offer this as a service.
Thank you very much, Ed. I'll be quick. As Ed has shown, the technology is ready, and we believe it can be very impactful. And as always, Schrodinger doesn't keep its secret sauce for itself, and it tries to give as much access as it can. So you can access this technology as a software, and the GUI is going to come out. But because it can be demanding computationally in terms of licenses, we wanted to offer it as a service too. All you have to provide us if you want to move forward with the service is the anti-target of interest. You need to pick among the ones that we propose, and some SAR. And to reinforce what Ed said, it doesn't need to be dozens of compounds. It could be a few compounds. Of course, there's a trade-off with how discriminating that's going to be. And we will deliver to you the most predictive model in around 3 to 4 weeks. The price is fully included. It takes a different discussion, but it includes computing, licenses, and FTE, and no milestones, of course, or things like that. So again, very accessible.
Thanks, Jeremy, and thank you all for watching. I think we have time for some questions.
So I can see a number of questions there. And do you want to kick it off with the first one, which is about I guess the GPCR study?
Right.
Is how did we know it was going to the pocket? The orthosteric pocket.
Right. That's a great question. Well, we effectively, let me repeat the question to be precise. How was the binding pocket identified for this target without a solved experimental structure? Purely ab initio by sitemap or using some prior knowledge from its GPCR family? In short, it was using some prior knowledge. So we started with the assumption, well, it's probably going to bind in the orthosteric site. So this was somatostatin. And there was, at the time we did this, there were no structures of any of the SSTR receptors, as we mentioned, but there was known the structure of somatostatin, the natural agonist. And somatostatin has a lysine, it's a peptide, and it's been known since, I think, the late 70s that the lysine is crucial for activity. Now, along those lines, we saw in the data series, in the ligand series, that there's a very conserved positive charge. So we engaged in the assumption that this is going to bind in the same place as somatostatin. Somatostatin was very strongly suspected to bind in the orthosteric site, so we went with that. Now, it's a very legitimate question because if you have no idea as to the binding site, this is going to be more challenging. Not impossible. You may have to guess, but you can imagine the computational cost is going to grow as you have to both, determine binding mode and determine the binding site. The fundamental assumption, though, is that with a reasonably sized ligand series, you can be proven right or wrong. So if you pick the wrong binding site, FEP should not show good recapitulation of the experimental binding affinity or functional data. And if you pick the right binding site and have the right binding mode, things should line up.
Okay. Maybe I'll take this one and we can alternate. Which model is more accurate between PDB and AlphaFold? I think this is a good question. Obviously, when there is an experimental structure, we prefer using an experimental structure. There are a number of things that are very specific to the target that AlphaFold might not be able to pick. But I also want to make the point that with the approach that we have, even if you have PDBs, it might be worth using AlphaFold as a starting point, because if the PDB is APO or if it's with a completely different series, it might not converge towards the pose you need for the series of interest. So experimental is always preferable, of course, but AlphaFold can be useful even in the context of a project that does have some experimental structure, if you struggle to get somewhere.
Sounds good. I see another question here. If you pick an arbitrary 3A4 structure, would it be much worse? Any data to answer this? So there were a number of places where we showed 3A4. One was we did this retrospective study using, I think it was a JMed Chem paper. Another where we had an internal program. The answer is, yeah, it would be much worse. So recall that we had thousands of models to choose from. And we ran FEP on a subset. You can recall from the subset that really only one model survived. Even independent of that, of the thousands of models, many of them, the vast majority of them, could only bind one ligand in the series. They couldn't support binding, another ligand or 2 or 3 ligands in the series. So the vast majority of the structures out there don't lead to something productive. So, what we imagine this as are sort of overlapping radii of convergence. So there may exist a small number of structures that are good enough for your ligand. And then within that small set, there may need to be small refinements that are necessary, some small side chain backbone motion. That's handled by using IFDMD. Then there can still be small variations in the series. Things are dynamical in real life. They're not static. FEP is using MD, of course. It has a finite simulation time. So there's a radius of convergence there. So between these overlapping amounts of sampling, we're able to narrow down and find something. But I don't want to describe it as a needle in a haystack, but you can't just pick an arbitrary structure, one structure, hope it's going to work. You're using these other structures and you're using all the techniques described here to cast a wide net.
Makes sense. I see we're out of time, but maybe we can take 1 or 2 more questions for people who can stay online. So one very quick, can we use FEP for free energy profile calculations of non-charged conserving processes, basically protonated to deprotonated state? The short answer is yes, absolutely. We developed the technology to do that. It was tricky, harder to converge, but yes, you can do that. And maybe one last one, because I think that FEP clarification is very useful, very important, which is the advantage of using FEP plus instead of umbrella sampling or MMPB GBSA. So I'll separate FEP versus MMGBSA, let's say. It's much more rigorous, right? You might have a good correlation with MMGBSA on a specific subseries, and it might work for you, but FEP is calculating all the terms, sampling both ends, and having a full thermodynamic cycle. And you have a number of papers out there that show that the accuracy overall is not comparable. Now, FEP versus umbrella sampling, I don't think there is any umbrella sampling implementation out there right now that can do the type of things we do with small molecule ligands. Umbrella sampling can be adapted for protein movement, things like that. But even there, FEP has shown some advantages. So these are technical differences, but right now only FEP plus or FEP is ready for big time, for small molecule. So shall we keep going? I'm fine staying and answering all these questions for people who can stay.
Same here.
Take another one. Let's do that.
Okay, I see one that I bet a bunch of folks have been wondering. Similar to the CYP% inhibition versus IC50, for the development of the hERG model, I guess the type of measurement also matters. What would be the ideal one in this case? A binding IC50 or patch clamp IC50? Choice is often limited when using historic data. Right, so that's a great question about the kind of data you want to use in FEP. In the prospective case, it was actually patch clamp IC50. Now, ideally, you would have binding data because that's directly what FEP is measuring. It's measuring binding affinity. The advantage, it's not prohibitive, though, to not have that. And that's the advantage of relative FEP, that the functional activity should correlate with the binding affinity. If you have that correlation, relative FEP should cancel out the offset between functional and binding activity. If that offset is constant across your series, relative FEP should cancel that out, and you can use that model just fine with a functional assay. Now, it doesn't guarantee that every functional assay is going to show this linear correlation across a wide activity range between binding and functional activity. We have lots and lots of evidence, and I believe there's tons in literature of where it correlates well. In the case of these anti-targets, though, that assumption becomes even more likely to be true. Put it this way, eventually, if the ligand is binding weak enough, it's not going to functionally activate or inhibit the anti-target. So at some point, the binding affinity is going to go to zero, and so is the functional activity. And so at that limit, they do agree, of course. So when you're trying to get away from binding, you have a very high likelihood of that correlation being valid, and there's still lots and lots of evidence of the correlation between functional activity and binding still being valid, even as you go in the other direction towards more and more potent things. But still, if you can, you'd prefer binding, but it's not a showstopper to have functional data.
Thank you, and I'll just take a last one, and then we'll end up there. But this one is, I think, can be useful. What are the best practices for determining when a homology model is good enough? Just look at the Ramachandran. So that's a good question, but I want to differentiate two levels. You have the level of the general structural quality of the system, and you have the level of, is it predictive with FEP? Is it a physically good model? We have a panel which is called Protein Reliability Report that looks at Ramachandran, but not only, right? You have a lot of flags that, a lot of things and red flags you can look at to see if a protein model is of good quality. You can have buried donors, acceptors. You can have clashes. You can have strain in the loops or things like that. So we have look at the protein reliability report. It lists all of them, and it's a really good tool to do that. Now, it's not because a model is looking good on the protein reliability report that it's going to be predictive for you. And I think this is a very important distinction. This is pretty much at the heart of what we're saying here. What's going to be useful for you is what empirically has shown to be predictive on an existing data set in the past. So what we're going there is going one step beyond a good model. We want a predictive model, and that's the whole point there. Yeah. So thank you, everyone. We went over time, but I hope it was useful, and see you on the next webinar.